mitochondria that signals apoptosis. Anybody? Cytochrome C, very good, excellent. Cytochrome C. So cytochrome C is a molecule. Basically what it'll do is that it will, once released, it forms a complex with a protein called APAP. What APAP stands for is the apoptosis activating factor. APAP will then proceed to basically activate another protein called caspases. So we'll talk about caspases a little bit later. But for now, just know that cytochrome C is what? It's a component of the electron transport chain, if you'll recall. You don't have to know that per se, but maybe you might encounter it in future studies. But cytochrome C is a member of the electron transport chain. It's in the inner membrane of the mitochondria. It's not supposed to be in the cytoplasm. So if there is some damage to the mitochondria and it's releasing cytochrome C into the cytoplasm, that tells the cell that something is not right. And usually what will happen is if there is significant damage, then the cell has to commit apoptosis. Commit basically programmed cell suicide, in which the cell, what will do is that it will proceed to activate these proteins called caspases, which are indeed the driving agents, the driving forces of apoptosis. One of the things that the APAP will do is that it activates these caspases, which in turn will have a variety of effects, which we'll talk about. But for now, just know that a cytochrome C is an example of an apoptotic activating component. It causes activation of apoptosis, and we'll talk a little bit more about apoptosis a little bit later. If I could direct your attention briefly to page 491 in the textbook, page 491, figure 15.7. So, when you have these caspases, what are caspases? Well, caspases is an acronym that stands for cysteine-containing aspartate-specific proteins. It's not necessary that you know that, but as long as you know that caspases are protein cleaving enzymes. They break down proteins. That's what they do. Caspases, they break down proteins. They break down the peptide bond. They're peptidases. Peptidases, if you learn biochemistry. Caspases initially exist in an inactive form. When one caspase is cleaved by another caspase, then the cleave then becomes active by aggregating into a complex, and they form this active executioner complex. The active executioner complex then proceeds to activate certain target proteins. For example, one thing it'll do is that it'll inactivate a DNA endonuclease sequestering protein. What effect will that have? Okay, so let's think about what a DNA endonuclease sequestering protein does. What does sequester mean? To withhold, to impede, to block, to prevent. So what the DNA sequestering, endonuclease sequestering protein is doing is preventing the endonuclease from going in and cutting down the DNA, which is something we would ideally not want, because that will lead to the destruction of the cell. But once this active executioner caspase comes in, it will break down that sequestering protein, doing what? Releasing that endonuclease, which will go in then and break down the DNA of the nucleus, which will of course lead to the destruction of the cell. Another target that it has is called actin. Actin is one of the cytoskeletal elements. It's responsible for maintaining the shape of your protein, among others such as muscle contraction, things like that, but it's responsible for maintaining the shape of your protein. They act like the girders that hold the infrastructure of the cell, the cellular skeleton, so to speak. If you break the actin down, then that will cause a loss targets for these cat spaces will include breaking down organelles, breaking down enzymes, things like that. Eventually all of this will culminate in the fragmentation of the cell. If you go back to 15.6, once you break down the cell, by breaking down, for example, actin filaments, things like that, the cell will defragment into what are called apoptotic bodies, and then apoptotic bodies are then going to be phagocytized by various scavenger cells of the immune system. It's a very clean process, apoptosis. Another process that we normally see that Dr. Brownson may have mentioned is called necrosis. Is, for example, if you were to prick yourself with a needle, then that damage to the skin causes inflammation of the tissue. What's actually happening there is that the cells they are dying, but they're dying in a rather gruesome way. And the cells release their contents to the extracellular surface of the environment, causing damage to other cells. And that results in various inflammatory reactions. But that's not happening per se in apoptosis. Apoptosis is a very clean process that one cell can end its existence without harming any other neighboring cells. But in necrosis, that's a completely opposite process, in which the death of one cell does impact other cells as well. You don't have to know about necrosis, but I was simply giving you sort of a contrast to help you better understand what apoptosis is. If you direct your attention to page 492, 492, there's an interesting paragraph that talks about how cytochrome C activates apoptosis. Cytochrome C binds to APAP, and then APAP, once it's activated, this complex